Dr. Madabhushi is the director of the Center of Computational Imaging and Personalized Diagnostics and professor in the Department of Biomedical Engineering and a lot of other departments. I'm not going to mention all of them. So he's pretty famous at Case. He's also a member of the Case Comprehensive Cancer Center. Dr. Madabhushi received his bachelor degree from Mumbai University, his master from University of Texas, Austin, in biomedical engineering, and also his PhD in bioengineering from University of Pennsylvania. He joined Case here in 2012, coming from Rutgers, and he has been a full professor since 2014. And the talk is to discuss the developments in computational image analysis tools for predictive modeling of digital pathology images for the detection, segmentation, feature extraction, and tissue classification perspective. So with that, I'd like to give welcome to Dr. Madabhushi. There are three.
Okay, thank you.
Thanks.
Thank you, Hadrian, for the very kind introductions. I'm very happy to be here and really talk about the work that my group has really been championing. I'm just sort of the conductor, if you will, who likes to stand here and take credit for the work. But this is really the folks in my group who've been doing the heavy lifting here. So I want to talk about deep computing for digital pathology, or how image computing can truly revolutionize precision medicine. So the major focus of our work is actually in the space of cancer. And the reason why this is such a critical problem is if you look at the statistics here, they're truly alarming. The lifetime probability that a male individual in the United States is going to be diagnosed with some form of cancer is 1 in 2, 50%, some form of cancer. For women, it's only marginally better. 1 in 3 women in the United States has a lifetime probability of being diagnosed with some form of cancer. So if you look at the totality of those statistics, that means 2 in 5 people in the United States are likely to be diagnosed with some form of cancer in their lifetime. While that is terribly unnerving, the fact is that the number of cancer mortalities in the United States has actually not been going up in a concomitant corresponding way to the number of cancers being found. That is, we're doing a better job with detection, early detection, diagnosis. We're doing a much better job in treatment because we're catching these cancers earlier, we're treating them earlier. But there is a bit of an imbalance here between the number of cancers we're finding and the number of cancers we're treating. In other words, we might, in fact we are, treating a lot of cancers that may not require aggressive treatment. We're treating with surgery, with radiation, with chemotherapy, very aggressively, a lot of cancers that might be candidates for active surveillance, for watchful waiting. So there is clearly a need for us to be able to develop tools, to be able to develop technology, to be able to reduce this issue of overdiagnosis and overtreatment of cancers. And with this sort of mandate in mind, the White House, and in fact, through the National Institute of Health, has launched this program on precision medicine, which is really this approach for disease treatment and prevention that takes into account individual variability in genes and environment and lifestyle for each person. Now, a big part of precision medicine is clearly big data or big data analytics. The ability to use computational tools in conjunction with all of this data that's being generated at a gene level, at a molecular level, and in fact, as I will present in this talk, at a tissue level, at an image level. The ability of these tools to be able to go into all of those vast swaths of data and to be able to prize out measurements, to prize out attributes that allow us to predict which of these cancers are more aggressive and therefore require more aggressive treatment, and which ones are the less aggressive, more indolent types of cancers that might benefit from either minimal treatment or perhaps from no treatment at all, just perhaps watchful waiting. Now, this notion of what is called companion diagnostics, this notion of tools, predictors to be able to figure out which cancers are bad, requiring aggressive treatment versus which cancers are not so bad and don't require that aggressive treatment is not a new one. So this notion of companion diagnostic tools, predictors, to be able to make these predictions, distinguish between the more aggressive and the less aggressive phenotypes, has been around. So a number of companies today, I'll mention one, Genomic Health, that has a highly successful assay called Oncotype DX, has actually been around for over a decade. It has a very, very successful set of products that focus on trying to predict which breast cancers are more aggressive and therefore require chemotherapy from breast cancers that perhaps might benefit from no additional chemotherapy. That particular test actually uses gene expression information, but there are other assays, other companion diagnostics that look at protein expression and protein level measurements to try to make these predictions. One of the challenges that all of these tests have is that they compute these measurements or prize out measurements about the gene level, about the protein level, within a very small area of tissue, within a very small region of surgically excised tissue or biopsy excised tissue specimens. And the problem with that is that it doesn't give us a comprehensive portrait of the biology, about the variability, about the attributes of the cancer. So one of the things that we've realized in the last couple of decades is this notion of intratumoral heterogeneity. We are now acutely aware of the fact that cancers that are very heterogeneous, that are complex, that look very different molecularly, phenotypically, tend to do very poorly. And the problem with assays that are looking at trying to make these measurements or make these predictions based on a small part of the tumor sample are perhaps not capturing the entirety of the heterogeneity or the variability of the tumor. But what if we could actually do that? What if we were able to take the entirety of the surgical specimen, the tissue specimen, and be able to comprehensively interrogate all aspects of that tumor? Could we now start to come out with a description of that tumor that accounts for this intratumoral heterogeneity, and by doing that, come up with a more comprehensive portrait, a more comprehensive prediction of the outcome of that particular cancer for that particular patient, and therefore be able to predict whether this patient requires more aggressive therapy versus not. And the major breakthrough that has happened in the last decade or so is this notion of whole slide digital slide scanning. So we now have whole slide digital scanners that essentially look like your traditional paper copiers. They only cost a little more, but not too much more. And they are able to take these glass slides, these tissue slides, and create high-resolution whole slide images of these tissue samples. And because they are now whole slide digital images, we now have the ability to apply big data analytics, computational image analysis tools to comprehensively interrogate the entire landscape of these tissue images. And what you're seeing here is an example of that. This is a high-resolution whole slide digital image of a standard hematoxylin eosin stained tissue specimen. But what you're seeing overlaid on this image are the results of the analytic tools that we have developed and applied to this image to be able to prize out measurements that capture comprehensively the description, the quantitative description of the morphology, the heterogeneity of the tumor. So we're able to actually go in at a per cell level, on a per nuclear level, start to look at nuclear arrangement, start to look at nuclear texture, look at the variability of the chromatin patterns in the individual nuclei, look at the variability and the morphology of the stroma and the epithelium, and really now start to get that comprehensive description of the variability of this tumor. Now, why is this important? This is important because there's fundamental information that's embedded in these tissue slides. And these tissue slides are, at the end of the day, a reflection of what's happening at the genomic level, what's happening at the proteomic level. So if we're able to comprehensively, quantitatively describe the appearance of the tissue morphology, then essentially what we're doing is capturing a reflection of the genomics, of the proteomics, but we're doing it in a way that does not that accounts truly for this intratumoral heterogeneity. We're able to capture the entirety of the complexity of the tumor and use these features that we're mining from these tissue slides to be able to now predict whether this is aggressive disease and therefore predict patient outcome. So here's an example of the kind of features we could prize out. So you can look at the individual nuclei within these tissue samples and look at nuclear architecture, and we know that nuclear architecture is highly correlated with disease aggressiveness and patient outcome. The more chaotic that architecture, the worse the cancer, and therefore the more aggressive the treatment that's required. You can look at the textural variations, the heterogeneity, like I said, within the stroma and the epithelium. And the beauty is with the big data analytic tools and digital tissue images, you can now actually do this across multiple different fields of view. And you can start to see that as we go out and look at finer and finer resolutions, the complexity of the features starts to increase. So what you're actually seeing is now a true manifestation of big data that's coming from these digital slide images, which is not coming from the genomics or the proteomics, but it's coming from the tissue level representation. And like I said, if you're able to capture a quantitative portrait of the complexity of the tumor at this tissue level, then effectively we're able to capture also what's going on or what's being reflected at a genomic and a proteomic level. So one of the challenges is, of course, when you're dealing with big data, you've got hundreds, thousands, potentially millions of structures, millions of cells, millions of nuclei, hundreds of thousands of glands. How do we actually go about identifying all of these in a high throughput way? Because clearly it's not possible to go in and manually identify each of these individual cells. So our group has been developing tools. Unfortunately, is the movie going to play? Unfortunately, the movie doesn't play. But if the movie did play, what you would have seen is that the individual glands can now be segmented within this prostate tissue pathology image. And using the architecture, using the shape and the morphology of the individual glands, we're now able to predict exactly how these patients might do. So we're not just looking at glands, we're also looking at individual nuclei. And I suspect this movie is actually going to play, so that's great. So I want to just do a shout out to Yu Zhao, who is a scientific programmer in my group, who's taken a lot of the algorithms we've developed in our group and has embedded it within this software tool that we hope to be able to make available to the community pretty soon. What you're seeing is a very high resolution digital slide tissue image showing hundreds of millions of nuclei that can now be automatically identified and segmented using our deep learning, deep computing algorithms. And by doing that, we're able to now capture tumor morphology and tumor architecture and nuclear architecture in a very, very comprehensive quantitative fashion. And just to give you an idea, this image you're looking at is several gigabytes in size. This is a 40,000 pixel by 40,000 pixel image with literally millions of nuclei embedded in it. But you can see with the blue spots on this image that all those individual nuclei, at least a majority of those individual nuclei, have been automatically identified. So how do we go about doing this? So one of the lessons that we borrowed is from the computer science, computer engineering literature, the so-called notion of deep learning, which really as a fancy way of referring to a cascade or an architecture of neural networks. So if you take all these neural networks and put them in a cascaded form, then you're able to create what the so-called deep learning, deep computing architectures. And this actually has been getting a lot of buzz recently. I'm sure a lot of you are aware of this competition that happened, I think just a couple of months ago, where Google went head-to-head with the world champion at this game called Go. And effectively, in a 5-match competition, Google's deep computing, deep learning framework was able to actually beat the world champion at Go 4-1. So we've actually been taking those ideas with deep learning and deep computing with this multi-neural network architecture to be able to apply them to questions in cancer diagnosis and precision medicine. So one of the questions we've been asking is, can we go and train the computer in this deep learning architecture to identify where cancer is? So here's an example. This is a breast pathology image with a region of cancer that's been annotated by our breast pathologist, Hannah Gilmore. Hannah went in and carefully annotated on hundreds of these images where she thought the cancer was, and we used this to then train the deep learning algorithm to be able to go in and identify cancer. And what was quite amazing was that the computer was not only able to learn, it actually started to beat Hannah. So what you're seeing here are little gaps where you see the probability map, which is what the computer essentially fleshed out as being the areas of cancer. You're seeing little gaps within that area that Hannah marked up. And at first glance, it seems like the computer missed certain areas of cancer, but it turns out the deep learning architecture got so intelligent, it was able to go and pull out small areas that actually did not have cancer in them. So given Hannah had a limited amount of time. She went in and sort of circled out regions that she thought were primarily cancer. But the computer was able to learn that there were specific areas within this that actually did not have cancer. So the ability of having tools like this is that you can now start to do high throughput things and sort of alleviate the burden on pathologists so they can focus on the more difficult, more confounding problems. But there too, I think the deep learning architecture and deep learning algorithms can play a role. And I've been talking about precision medicine, and I want to sort of very quickly touch on a few use cases where this deep learning, deep computing architecture can actually allow us to address questions about who to treat and how to treat. One of the problems we've been looking at for a while now is ER-positive breast cancers, the most common type of breast cancer. There is 130,000 women in the United States every year are diagnosed with this form of breast cancer. And the principal conundrum with the diagnosis of these cancers is who to give chemotherapy to and who not to give chemotherapy to. Because it turns out that 80% of these cancers will actually benefit from what is called hormonal therapy alone. They don't require chemotherapy. It's the 20% that who have the more aggressive phenotype who require chemotherapy. Now, like I said, Genomic Health has had an assay called Oncotype DX that looks at 21 different genes and is able to make a prediction about who has the more aggressive disease and therefore requires chemotherapy from the ones who don't have the more aggressive disease. But turns out that this is an expensive test. Genomic Health's Oncotype DX is $4,000. It's a tissue destructive test. It destroys the tissue. And it's really not feasible in areas outside the United States where you can't physically ship tissue because this test requires shipping of tissue. About 9 years ago, Sridhar Ganesan, a breast oncologist colleague of mine, and myself sort of came up with this idea of the image-based risk score. So if we were able to digitize tissue images of breast cancer, could we then interrogate it with the analytic computational tools and come up with a prediction of who has more aggressive disease and who has less aggressive disease? And this gave birth to the idea of IBRIS, or the image-based risk score. The beauty of this, of course, was that it was non-destructive, and we could come up with a prediction for pennies on the dollar as to who might have the more aggressive phenotype and less aggressive phenotype. And of course, because this was digital, you could take advantage of sort of a cloud computing architecture and not actually have to ship physical pieces of tissue around. One of the breakthroughs we've had recently has been to look at new ways of assessing nuclear architecture. And towards this end, we've borrowed an idea from chemistry called the Hosoya index. And so the Hosoya index has been around for a while, and it's been used to look at the complexity of chemical compounds. And we realized that that was a powerful idea because it allowed us to look at nuclear arrangement. And like I said, nuclear arrangement has been shown to be highly correlated with disease aggressiveness and patient outcome. So I'll skip the details about how the Hosoya works, but effectively it looks at cycles in a graph and tries to figure out multiple permutations and ways in which you can go from one node on a graph to other nodes on the graph in multiple different paths. But we were able to use this notion of Hosoya index to now look at nuclear architecture in patients who had breast cancer but early recurrence. That means the disease, they were treated, but the disease came back, and therefore they had more aggressive disease. And we also looked at distant recurrence patients, so patients who were treated, and the disease did come back, but it came back very, very late. So effectively, they had good outcome or good prognosis. Working with David Rim at Yale University, we were able to cull together a learning cohort, a learning set that was used to build our model. And we also had an independent validation set. And this is critical when you're developing algorithms like this. You've got to make sure your learning is separated from your validation. So we had 210 patients where we were able to validate how well the model was doing. And these were the results. So we presented this at meetings. We've yet to publish this data, but it's something I'm very, very excited about. And what you're seeing here are the Kaplan-Meier curves, or the survival curves. And what the main takeaway from the curves is that they're separated. The model is separating out patients who had early recurrence from distant recurrence. And our overall accuracy was 76%. Now, to the computer scientists in the room, that might not look like a great deal. But to put this in perspective, this $4,000 Oncotype DX test has been reported to have an accuracy of about 64%. So effectively, by just looking at the tissue images, we appear to be able to do much, much better than sort of the gold standard. And why is this important? I think I've sort of touched on these issues already. This is a very, very low-cost test that really could have impact in lower middle-income countries. It's a very rapid test. You could essentially do the test in orders of magnitude less time than the current gold standard Oncotype DX. The tissue is digitized, not destroyed. The accessibility is worldwide, not just in the United States. So effectively, you have an inexpensive, fast, reliable, accessible prognostic breast cancer test that's priceless. For everything else, there's MasterCard. But the implications of this go beyond breast cancer as well. So we've been looking at a number of other cancers. One example is lung cancer, and just a shout out to Shangshui Wang, who's my graduate student who's been working on this project. And working with Dr. Vamsi Valchetti at the Cleveland Clinic, we've shown that in early stage lung cancers, these tools could actually allow us to predict, again, which of the early stage lung cancers have early recurrence versus distant recurrence. And you can see, again, from the separation between those curves, that the test is doing a very, very good job. In fact, we're about 85% accuracy in predicting which early stage lung cancers might have early recurrence versus distant recurrence. And this is critical because, again, the issue is who to give adjuvant chemotherapy to and who not to give adjuvant chemotherapy to. And we've been taking this idea to other cancers, looking at oropharyngeal cancers, which typically tend to have good outcome. But we've realized that if we look at the nuclear architecture within different tissue compartments, looking at the stroma and within the epithelium, we can build predictors that can tell us exactly which of these cancers will progress on to metastasis and which ones will not. And we actually published a paper in American Journal of Surgical Pathology a couple of years ago where we showed that we could actually do this prediction significantly better than a number of the pathologic variables such as T-stage and nodal status. So we could do better than is currently happening right now in the pathologist's office. We've taken this idea and applied it to other cancers as well. Prostate cancer is another example, which obviously is of extreme interest to all the men in the room. And we've shown that if you look at glandular architecture, the way glands are arranged, if you look at the disorder of these glands, these gland orientations, they tend to be more disordered in those cancers that are more aggressive, prostate cancers that are more aggressive, versus the prostate cancers that typically tend to have good outcomes. So we found a way of mathematically modeling this disorder in this architecture and shown that using this idea of disorder in gland orientations, we can actually predict which prostate cancer patients have failure or disease recurrence after surgery versus those who do not. I'll just skip on to this next slide and sort of just do a quick shout out to Dr. Pallavi Tiwari, who's a freshly minted assistant professor in biomedical engineering. Dr. Tiwari is also affiliated with the center, and her interest has been on GBMs, on glioblastoma multiforme, and she's been looking at MRI. But effectively, this idea of deep analytics can be brought to bear on MRI scans of brain tumors as well. And what she's shown is that if you look at features of these tumors, not just within the tumor, but outside the tumor, those features outside the tumor appear to be highly predictive of how these patients will do. And that's very counterintuitive because you would imagine you need to look at the tumor area. What Dr. Tiwari has found is that it's the features outside, immediately outside the tumor, that seem to tell us a lot about how these patients will do. And she's actually shown that she can statistically significantly predict which patients will have longer-term survival versus which patients will have shorter-term survival. And this is some of the data that she presented at a meeting last year. So let me just conclude with that idea. So hopefully what I've conveyed is that image analytics on medical imaging and digital pathology could help address questions in precision medicine. Image analytics can work with standard of care imaging in a non-disruptive, low-cost, accurate way, and helps to address questions about who to treat, how to treat, and more importantly, did the treatment work? Number of different projects in our center, number of different cancers, and I've just touched on some of them. We're looking at prostate cancer, breast, rectal, lung cancer, head and neck cancers, brain tumors. So hopefully what I'm conveying is that this idea of sort of deep computing with digital pathology really lends itself nicely to a number of other, number of cancers, number of other diseases as well. Like I said at the start, I'm just coming in and trying to take credit. These are the guys who do all the work. And just a shout out to the outstanding folks at the Center for Computational Imaging and Personalized Diagnostics. We've grown to over 30 people in the last 3.5 years at CASE. It's been an outstanding ride these last 3.5 years. We've been very, very fortunate to have funding from National Institute of Health and the Department of Defense and a number of other centers, and continue to sort of build on our initial successes and keep going. Thank you very much.
If there is any question, feel free to walk up to the microphone and do the questions.
Thank you, Anant. This is really exciting. How far in time are we from having these tests actually used in medical treatment?
Thank you. That's a great question, one I get asked quite a bit. So we've actually had some very good news recently from the National Cancer Institute. We were just told that a academic industry partnership grant that we wrote on the breast cancer work is going to be funded. So this is a 5-year grant where we're going to be working with Inspirata. Inspirata is a cancer diagnostics company out in Tampa, Florida. And we're going to be working with them to take the algorithms, the tools we've been developing in the lab, and create a prototype, essentially a pre-commercial prototype, which will then be translated and make commercially available. So this is a 5-year grant. Our hope is that in sort of the next 2 to 3 years, we will have the prototype. We can start to do field testing. One of the things we're thinking about is immediately targeting this in lower middle income countries where they just don't have any test, $4,000 or otherwise. So we think that we can make a play for sort of those LMIC markets. And that's and the good thing is that we have this grant, so we are going to be working towards sort of realization of that. My hope is that we'll have a prototype in the 24-36 month time frame, and this can be deployed and start to be used in that time frame. Yes.
Can you elaborate on what you mean by nuclear architecture?
Sure. So you have individual nuclei in tissue images. The nucleus is, of course, sort of the core element where all the processes, the energy for sustenance is derived. So the tissue pathology images have essentially all of these various nuclei, and different diseases or different grades of disease show different architectures with that arrangement. So we know, for instance, with breast cancers, pathologists will tend to look at something called nuclear pleomorphism. So how variable are these nuclei? If they see that the nuclei are highly variable, then that's a bad sign, because that means your nuclei are very pleomorphic. They vary in their shape, their size. And that's essentially a sign, a red flag, if you will. This is highly aggressive disease. Pathologists tend to do this visually, but the problem is that pathologist grade tends to be variable. So we have an outstanding breast pathologist, Hannah Gilmore, who I mentioned here at Case. But if you go around and look at community pathologists or non-specialized pathologists, there's a lot of variability between what pathologist A might say is high grade or highly nuclear pleomorphism versus someone else who might have a different take on it. So the problem is that the subjectivity of pathologist interpretations can confound what our prediction is about the aggressiveness of disease. With tools like this, we're able to quantitate the process and therefore have highly reproducible measurements that are not subject to the same sort of variability that human readers might be.
If not, thank you so much.
Thank you. Thank you very much.
What an awesome way to open the session.